to start. So I'd be pleased to introduce Betsy Siewert, who's going to give our presentation on evolution and relationship among the species.
So I'm going to be talking today about evolution and the relationship among the species. And I wanted to start with saying that I'm going to argue that the species are related, and people have been wondering and trying to categorize how they are related for thousands of years. They started by grouping the organisms based on similarities and differences. For example, Aristotle, almost 400 years ago, had organized the species, and he split it up between plants and animals. And then under plants, he said, well, there's some that are trees and some are shrubs and some are herbaceous. And animals, there's some with blood and some without blood. And then those with blood, those are some who bear their young live, and then there was egg bearing. And then he split up the ones without blood as crustaceans, mollusks, and insects. So people have just been trying to look at the species and how are they related with each other. Theory of evolution, in a nutshell, is that the life began in a warm little pond, according to Darwin, Charles Darwin, and that it had nutrients in it, and the nutrients, along with some sort of energy, maybe coming from lightning or something like that, started putting some of these nutrients together to make biomolecules. And then the biomolecules would eventually join together to make the first cell. And so he thought that this was maybe how life had started and how we got all the species. So in a nutshell, the theory of evolution, according to him, is 3 points. It's natural selection based on random variation. And then there was that common ancestor that was in the warm little pond. But before we start the whole explanation of that and discussion of that, I wanted to defend Darwin to begin with. When he was thinking about life, he knew that there were cells, but this is what he knew about the cell. They could see the nucleus, they saw the nucleolus within the nucleus, but they thought that the life was very simple and easy to develop. This was in the 1800s. They did not know that the cell really looks like this. The cell is more like a city. It has a protein manufacturing plants, and then those proteins are sent to a processing plant, which gives it a tag and sends it to the post office, and then that will direct the proteins to go where they're supposed to go. There's power plants, there's waste disposal plants, and most importantly, right here, I think you can see my cursor, is the information center. That's the nucleus, and it's not a simple little nucleus like what Darwin thought. It's got lots of different micro machines that are in there. And so that is the controlling center of everything within the cell. So when Darwin was developing his theory of evolution, he didn't know how complex the life really was. So we'll go back to what he was saying is that, natural selection based on random variation and common ancestor. I'm going to briefly go over the first two, and then most of my talk will be on that last point. So natural selection. I would say if the lion is chasing the zebras, he's not selecting the ones that are going to survive. It kind of sounds, according to Darwin, that the ones that survive are the very best ones. But when the lion is chasing the zebras, he's not going to look and say, oh, I'm not going to get that one, I'll get another zebra. He's going to get the weakest ones. So instead of having natural selection, I would say that it's more like natural elimination. The weakest ones die, but those that are in the middle all survive. So it's not selecting the best, it's just getting rid of the worst. Random variation is the next point. And this is one that I don't have any problems with. The random variation happens all the time. Here's a kind of a sad example of random variation where elephants are poached and killed quite often just for their tusks. And there's been mutations of variations where the genes that coded for the tusks, there were some that were broken. And so there's elephants now, there's more and more elephants that don't have any tusks because they can survive now because the poachers don't kill them. Most variation are mutations that break the genes. And there's a whole area there. Dr. Michael Behe has talked quite a bit and written a book called Devolution that talks about that genes, there's not much gain of function in the random variation. Most of it breaks genes to survive. The last one is common ancestor, and this is where I'm going to be spending most of my time. And rather than having a common ancestor, I'm going to argue that the species are related. And I'm going to talk a little bit about how, again, going more into detail about how they're related and why are they related. So initially, like Aristotle, people started trying to figure out how species are related. How do they look? The wolves here, here's the evolutionary tree, and they put the dogs close to the wolves because they look a lot alike. And then what do they eat? Do they eat plants or animals or both? How are you going to categorize these animals that you're looking at? And how do they get around? Do they fly, swim, or walk, or do they have 2 or 4 feet? And where do they live, on land or in water, or both? So you could draw these trees just based on what you can see by the way the animals look and act. But there's problems when you're trying to draw the tree. For example, bats, are they a bird or are they a mammal? Because they have wings, but they bear live young. And whales, are they a fish or animals that live, that give live birth? And what about animals that can live on both land and water? Are they fish or land animals? So all of these things were difficult to figure out. So there was a lot of interest in how do you draw these trees. So here's examples of 3 trees. This one on the left here was drawn by Charles Darwin. This was in his notebook right after he got back from Voyage of the Beagle. He went on this around the world trip collecting specimens and taking notes on evolution, or how he eventually developed the idea of evolution. But he had those ideas initially. And this number 1 here is the common ancestor. He was already thinking that there was one common ancestor in that warm little pond. And then things branched off, and this is where you got all these species branching off from that one common ancestor. And all of the trees, that phylogenetic trees that are shown, these are pulled out of different biology books that are textbooks. And again, it's always a tree. There's this one common ancestor here. And one thing I want to point out is this one right here. This branch are the archaea. Now there's 3 kingdoms. Go over to this right tree here. There are 3 kingdoms. Eukarya are pretty much everything you can see. Where you're part of the eukarya kingdom, plants, animals, pretty much everything you can see are eukarya. And then there's the archaea, and then the bacteria, which you know. The archaea are very primitive. They were just discovered in the 1970s, and they thought initially that that was maybe one of the primordial organisms that came out of that warm little pond. They're very weird little organisms. They look like bacteria, but they live like in icebergs. They live in hot springs. They live in high sulfur springs, in ocean vents. They just have occurred all over the place. And so they thought, oh, that's the original. So in this tree, they have put it right next to the root because they thought this was the primordial organism that came out of that, or close to that organism that came out of that warm little pond. As technology advanced, they realized that they could do more than just look at how the animals looked or the species looked. They realized they could do molecular sequencing. They could learn how to sequence proteins in the 1950s, and the DNA and RNA they were able to sequence in the 1970s. And so in case you're not familiar with these terms, DNA is that's what makes up the genes. That sits in the nucleus, and that is information for all the proteins and all the activity that goes on within the cell. That DNA gets transcribed into RNA, which is essentially a single strand, except rather than a T, it has a U, but essentially they are both nucleic acids and some differences in how they're put together. And then the proteins are made from each of a triplet of RNA translates into one amino acid. And so the proteins are made and they're read in the ribosomes 3 bases at a time to put the amino acids together. So that's what those molecules are. So they thought, well, if we can look at the sequencing, maybe that would give us a better idea of what that tree looks like, because it's maybe tells you more than what the animals look like. So then they thought, okay, let's try that. So here is the phylogenetic tree is on the left here. This is one protein that is shown here. It's color coded, so you can see two things that I want you to see on this. First of all, this is the tree from humans through chimps, through cows and horses, all the way down to fish and lampreys. And so the one thing that I find noticeable is that how much similarity there is across all of these organisms. The other thing to notice is, for example, here, where there's a little bit of grayness here and here, that's how this branch here got pulled off. Now, the other thing is this is just one protein. So if you look at other areas of the genome or other proteins, you're not going to get exactly the same tree. And so that's part of what the problems are, is that depending upon what you sequence, if it's a protein or if it's a DNA and an mRNA, for example, here, you get two different trees depending upon what you're sequencing. Here you can see that on this side, the rat and the mouse come off first, and then the guinea pig, and then there's the man and the elephant here. On this one in the DNA, they have the tree coming off the elephant first. The man is close to the elephants, the cow and the dog are in between. And so there's problems with this molecular sequencing. This is a paper that was published in 2003 in Nature, and this is a biostats paper. So these are all sequences of 7 varieties of yeast, all done in different ways. So you got all these different trees, and an area in biostatistics, there's a real big area of research of how to put all of these trees, phylogenetic trees together to get the true phylogenetic tree. How do you weight each of these trees? And that's an active area of research right now. So then the next thing they said was, well, maybe if it depends on which one you can, part of the genome you sequence, you get a different tree. Maybe we can sequence the whole genomes. And again, that was a result of technology, because the sequencing got to be really, really fast. It took a long time back earlier. People would do a whole PhD. Their thesis would be sequencing one gene and that would be their thesis. But now you can get things sequenced within a day or less to sequencing. So they were able to sequence whole genomes, and they thought maybe we'll get a better look at what the tree looks like. Well, this is what was published in Science in 1999. Again, this is a high-end journal, and they got what they called the web of life. And here's your 3 kingdoms. The other thing to notice on this is that it doesn't look much like a tree. And for example, here there's something from the bacteria that went over into the eukarya. It doesn't come from the branches down here, it comes across. And so when they don't understand something or they just want to characterize it, they give it a name. And so this is called horizontal gene transfer. And there's another one there. So then they thought, well, now what do we do? And so as technology carried on further, they were able to sequence genomes without culturing them. The reason for that is the advancement, what they used to do to sequence a genome or the RNA or whatever they're sequencing, what they had to do is grow the organisms and then extract the DNA or RNA and extract it and then culture it, or culture the organisms and then extract the DNA from it and then sequence it. And they developed a way of looking at this. This is in the 2016, that they actually were able to sequence whole genomes without culturing them. So they thought, well, we can pick up some organisms that maybe are missing that you would see. You know, you wouldn't be able to culture them, but they're there and they would explain more about the tree. So then they published their findings in Nature in 2016. And again, it doesn't look like what they're expecting to find. Now, the other thing I wanted to point out about this is, if you remember back earlier in the slides, I said that archaea were thought to be those very primitive organisms. They were just been discovered. And they thought that, oh, those are the first ones that came out of that warm little pond, or just recent descents from those. But what they found out in 2016, here's the archaea right here, and the eukarya are right here. They are right next to each other. The reason it is that the archaea have almost the same biochemistry as the eukarya. And so that was a real surprise, because they were expecting that to be one of those primitive organisms, and it actually is quite advanced. And so these are the bacteria all up here, which are all very different. Here's the eukarya, which is us and plants and animals. And here are these things thought to have been very, very primitive. Well, they have pretty advanced biochemistry. So then they thought, okay, now what do we do? So over the years, they start out with thinking it would be simple, that we're just going to draw that tree and we'll look how that looks and put all the things together. But in the meantime, they start seeing more and more complexity and more difficulty in the trees that they were trying to draw. Okay, so that's kind of a background of how they were trying to put phylogenetic trees together. So now I want to look a little bit about why are the species related. So according to Darwin, it's a common ancestor. This is a quote from his book, Origin of the Species, and it's: the small differences distinguishing varieties of the same species steadily tend to increase till they equal the greater differences between species of the same genus, or even of different distinct genre. So he was thinking in terms of that simple tree that he had drawn in his notebook. Another possibility is a common creator. And I just, for fun, I took about 6 pictures of the artist Monet, and I put them together and wanted to compare it to one of the earlier trees that I showed you. So here is an evolutionary tree assuming common ancestor. Again, all you can see, all they knew about were these final animals. This is all conjecture down here. And so they said through time, it starts with one branch and it keeps branching off. The dogs are closer, and so there's a more recent branch there. The cat's further on, so it's a longer branch. And so that was the way of building a tree using common ancestor. But you can build that same tree using a common creator. So here, these 2 paintings of Monet are very close. So I put them over on this side of the tree here, where they're very close. They have the same coloring. This has kind of the same coloring, so they were over a little bit further away. But you could say that, oh, well, they branched off from that. And then this painting here is very different. So I put that on the long branch. So if all you know is what's on top here, or the animals, you can build a tree, but that doesn't mean that it is a tree. So we are always assuming that it is a tree, but is it really a tree? common ancestor, that idea of a common ancestor requires it to be a tree because of this warm little pond. There was one one organism or one primordial cell, and it grew. And so you have to have just a single base, and it has to look like a tree and then slowly branch off. You can't have more than one base because this pond is just the one earlier ancestor. They call it the last universal common ancestor, Luca. So I'm going to argue that it doesn't look like a tree. So what does a rat, a primate from Africa, a tenrec from Madagascar, an opossum from South America, a lizard from Central America, a frog from Africa, and a bat from North America have in common? Well, they have 3,000 base pairs. Each one of these animals has 3,000 base pairs that are 96% identical. And this was published in a PNAS paper, which again is a very high-end journal, in 2008. And what they concluded was that analysis of spin transposons, the spin stands for space invaders, because they didn't understand how this 3,000 base pairs of DNA got into all these animals. It says analysis of spin transposons across species reveals a combination of features that is incompatible with the traditional hypothesis of vertical inheritance. In other words, vertical inheritance is that tree. It's incompatible with that. Why did all these animals from all over the world get the same sequences? Here's another PNAS paper, and it's there's whales. They looked at whales and bats and found that they have identical mutations. Surprisingly, these very different species evolved. They're still talking about it being evolved. Their unique ability to use sound waves to navigate and identify obstacles and tasty morsels, be they mosquitoes or minnows, in part by acquiring identical mutations in their genomes, mutations not shared by other more closely related species. So what they're arguing for is that there was mutations that helped to increase and develop the ability to have echolocation, where they can send out a signal and find out where the prey is. But they developed it each individually, but they both came down to the same way of solving the solution by developing this echolocation and having it do the same mutations in their genomes. And again, since they couldn't explain it, they put a title on it and they called it convergent evolution. In other words, that they both converged on the same solution in the same way, even though there was no relationship between each other and there was no other species around that had evolved that way. So those are problems for arguing for an evolutionary tree. So Winston Siewert developed another model for how the species are related, and he called it a dependency graph of life. Winston got his PhD in computer science from Baylor University numbers of years ago, and he published this idea in Biocomplexity in 2018. And what he said was that maybe the species are related not by a tree and slow evolution, but it's related, the species are related because they look related like a computer program might have different subroutines. And when you're programming, you don't always rewrite the same code. You take a code from someplace else that you've already written and put it into your code. And so I devised a picture of how this would look. So there's live birth and laying eggs, that's two ways of giving birth, and those with wings and legs. And then what I did is I took 4 different categories. The garter snakes are reptiles. Here's a couple of fish. There's birds, and there's 3 mammals. So who gives live birth? Well, it's the garter snakes, which is a reptile, guppies, which is fish, and the bats and cows. And then the other fish lay eggs, birds lay eggs, and a platypus lays eggs. So that looks like you would have taken let's get the live birth program subroutine and put that into the garter snakes and the bats and cows, or the same with the wings. The birds and bats have the wings, and it's like, oh, let's use that subroutine. And same thing with legs. And so what he did was he actually did, after he took, I think it was a couple hundred different sequences in a database that they have these available online. And he tried to put a tree together with using an evolutionary tree concept. And then he also put the tree together using a dependency graph. So it didn't really look like a tree. It just looked like a object-oriented programming concept. And he ran statistics on it, and he found that the dependency graph fit the sequences, and the model fit a lot better than the tree fitted. So is it a common ancestor, or is it a common creator? So we'll leave that, and we'll come back to that at the end. But I think that what I wanted to also say is that the species are related. How are they related? Well, I think it looks more like a computer program than a tree. Evolutionary theory says it's a tree, but they are related. And so there is useful things about what we'll call the evolutionary tree, but there's useful things about the species being related. So here is a paper that's published on genetics of alcohol-induced behaviors in Drosophila. Drosophila are those little fruit flies that when you leave your bananas out too long, they have the little flies. Well, those little flies are a genetic organism that is used in genetics labs. They use yeast, they'll use the flies, and they use a little worm called the Serranorhabus that they use for genetic research. So what they did with the flies is that they would put the flies in a little test tube, and they like to hang on a post up in the top. They don't like being on the bottom of the test tube. Then they would put pump alcohol fumes into the test tube, and the flies that were most susceptible to alcohol would fall to the bottom of the test tubes. They would take those out, breed them. Pretty soon they found there were some flies that were extremely resistant and some flies that were extremely sensitive. So they published papers, and I just pulled out a little bit from this paper to show you that geneticists do have sense of humor. Two of these mutant strains of the flies were named Tipsy and Barfly to reflect their increase in reduced sensitivity to alcohol, respectively. Then they found another gene, a mutant that shows increased sensitivity to alcohol, and it was called Cheap Date was the strain. And so why are they interested in alcohol sensitivity to flies? Well, that's because what they can do then is look for those genes that are in flies and look for them in humans and find out which genes affect alcohol sensitivity in humans. And that's only because the species are related, and you can do some biochemical work in flies and find out interesting things about humans. Another example is yeast. This is a picture of a yeast that's just about ready to divide. The blue are the chromosomes, the yellow are the microtubules that are going to be pulling the chromosomes apart, and on each end is a spindle pull body, which in yeast it's a spindle pull body. In humans it's called a centrosome. It's a little bit different, but it's the same idea. And this actually was a gene that when I was working in molecular biology in Boulder, the lab worked on MPS1. which is called monopolar spindle because it only had one spindle. There was something wrong when this gene was not working right. And so they found out, our lab found out that it was a kinase. And what happens is that when the cells divide, there's a editing process of kind of a checkpoints to see if everything's set up right and if it's all set, and if it's okay, it'll let it divide. If it's not okay, it just stops, and it makes the cell stop, and it goes into almost a self-destructive mode. There's a lot of checkpoints to make sure things divide properly. And when MPS 1 doesn't work properly, it starts to go crazy and make multi-cells with multiple, instead of 1N or 2N a number of chromosomes, they'll have 4N or 8N or 6.5N. They just start multiplying, but there's no checkpoints on it. And it turns out this MPS 1 is one of those checkpoints. So what they did then, we worked on it in yeast in Boulder, but later it was sent out and was published, and other labs looked at it. And what they did is they took the yeast that had a mutant MPS 1, it wasn't working. Now in yeast, when you're wanting to do genetics on it, you can have a gene that is kind of broken. If it's growing at normal temperature, it'll work. When you raise the temperature up to a high temperature, it doesn't work anymore. So they're called temperature dependent mutations. So what they did is they took the MPS 1 temperature sensitive yeast and grew them, and then they threw in a bunch of human genes into this mixture, and they raised it to a temperature and found out which human genes rescued this MPS 1 kinase that wasn't working properly. And that's what this paper is that this was the MPS 1 kinase in a kinetochore. That tells me that's a human cell's attachment during mitosis. So they found that this gene in humans was involved in making sure that there was everything was right with the cell before it was divided. And our lab, a part of our research in the lab that I worked in was funded by American Cancer Society because it was very much interest to cancer people, why cells divide or not divide properly. So 3 main points of this talk. I think I've gone through this pretty quickly. 3 main points of this talk, then going back through this, is that one is blind evolution and there's intelligent design. So in blind evolution, there'd be natural selection or natural elimination. Random variation is the same, except in reality, it's not, most of the time the genes get broken. It's not an evolution, it's more of a devolution, to quote Michael Behe, Dr. Behe. And the common ancestor, it looks more like it's, is it a common ancestor or common designer? And again, at this point with the trees, they only knew what was on top. They didn't know all the complexity underneath. And so they thought that it seemed to make sense that this could be common ancestor. But as they developed more and more technology, they realized that it probably wasn't like that. And the last point is, or the next point is that it's blind. Is it blind evolution, or if it's an intelligent designer, sort of like a programmer? Does a tree look like this, or does it look like a design program? I would argue that it looks more like a design program, and that the tree is shown not to be quite as well supported by the data as a design program is. The last point I want to make, though, is that even if there is a common creator, the species are related, closely related in certain aspects, and we can use that in research to find out things about humans by looking at experimental organisms. And why are they related? I would argue that there was a common creator. He didn't completely recreate things that were already working in other organisms, and so that he could look at kind of like a programmer using subroutines or object-oriented programming. So that's what I wanted to say. I hope I didn't go too quickly for it, and we can open it up to questions.
Okay, the question that we have right now is one of the criticisms of common descent is the lack of transition fossils even after hundreds of years of searching. Does this sort of a common creator eliminate that problem?
I would say not necessarily, because there was always the missing fossils argument. I think that when you look at common creator, I think people sometimes think about common creator that everything is created separately, but when you look at it, when you look at that they are related, you can look at it for that it was redone, that the creator reused some mechanisms in there, some programs, and just put it in the new organism. So the missing fossils, it seems like that is, if it's missing, they're saying, oh well, we haven't found them yet, is what they'll say, but they probably are missing because they weren't there. Does that make sense?
Yeah, it does. How does this relate to the vitamin C, the broken vitamin C gene in humans and chimps?
I haven't looked into that a lot. I think that people are looking at those things. There's also the chromosome 2 that in humans looks like it came from 2 chromosomes in the apes, and there are people who are working on it. I've heard some things about it, how they can put that together. The vitamin C, what they're looking at a lot of times is there, it looks like it's just broken, but is there a benefit for having broken it in humans that they don't know about yet, or they're looking into?
Okay, another question is horizontal gene transfer, is the horizontal gene transfer a hypothesis that is really more adequately explained as the reuse of functional routines by an intelligent designer?
I would say so, and I think there's a lot of evidence for that. What they argue, the evolutionary people argue, is that there is a piece of DNA that hops around. I forget what they call it, but it hops around into different organisms and inserts into those other genomes. But that seems like a just so story to me. I think it makes a lot more sense that there is a common creator who just reused the same mechanisms in different species.
Okay. Anybody have more questions? What is currently explained? What is the currently explained mechanism by which this gene transfer happens? I think the question is, how does horizontal gene transfer works? And I know there are sort of 3 different methodologies, but I can't bring them up from memory, but I bet you can, Betsy.
I don't know. I think one of them is that it's like a virus that pulls out a piece of the DNA and then jumps over to another organism and inserts it. So
yeah, there are viral transfers. There is actually a transfer via pili, which is an appendage that is made to directly transfer material from one. You see this in bacteria, one or the other, and then there's a third method, and I can't recall what it is right now.
yeah, the pili are actually, that is in bacteria, and that's a lot of ways they exchange DNA a lot, and that
Yes.
happens a lot in bacteria, and that's why when you get a bacteria that is antibiotic resistant, that's a problem because they transfer that in resistance to other bacteria. And so they're pretty prolific on sharing their DNA with other bacteria. But I
Okay.
don't know about higher organisms. I think the virus, it would have to insert into the DNA, and it just seems more awkward. I don't
Yeah.
think any of that, I think most of that's been hypothesized.
I know from my own work that there is horizontal gene transfer that takes place between the bacterial and the plant kingdom. That's something that's only been discovered and really verified in the last 3 or 4 years. That was not something that was expected. Another question that we have here, a couple of questions more coming in. Let's go back here. Where do orphan genes fit into this?
Orphan genes started out, they're spelled ORFAN. So it's ORF is open reading frame. So what it is that you have, if you're looking at this phylogenetic tree, and all of a sudden it has a gene, an open reading frame that doesn't have any predecessors. Sort of like what I was talking about with the what was it, the different animals that all of a sudden they just got that gene, even though they were spread across all those continents. And where did they come from? And they are, again, that's a time area of research right now that they'll say, oh, well, that just happened. And so I'm not quite sure what the research is on that.
Okay. Another question is, can such a virus work across wildly? And I think you're talking about in the case of, this is Sean, and clarify if you need to, can such a virus work across wildly different species like fish, bacteria, birds, and mammal? Sean, are you, and you can unmute to answer this question, are you talking about horizontal gene transfer? Can it work across species?
Yes.
Yeah.
That's basically it. If you have bats and whales doing this same feature, how did that get from one to the other? Obviously, they don't really interact, so you can't really explain horizontal gene transfer there. So you'd have to have independent development of this capability of, evolution of some species special feature twice. It's amazing, double amazing.
Yeah, that's why they give it a different term. It's called convergent evolution, because you can't get it from bats to whales very easily. So they call it convergent evolution. And they come up with those things a lot of times. They'll have, what was it? Something with snakes that they had one doing something and then something further removed from that. There's more than just the whales and the bats that have the phenomenon that they both develop the same thing in the same way, but independently, because there's no relationship between the organisms that have developed whatever these genes are.
Okay. So another question is there any acknowledgement of this thesis that, going back, I would think the dependency graph would be a good outside of the ID community. I mean, are people that are not ID people looking at things like this and, being honest and scratching their heads?
I think, at least when I was working in molecular biology at CU, I think that they just assumed it was true. They didn't really look, and they didn't care, because the important thing is the species are related. And most molecular biology research is done based on the species are related. The why and how are they related, that's what they were taught, that it's evolution. But I don't think they really even care if evolution is true, because that's not important to them. That's the fact that the species are related is what's important. And they just don't even question it, because it's not important to the work that they're doing.
So is another question was the similarities in echolocation in bats and whales conventionally explained as two independent developments of this capability through parallel evolution? We assume that these two species did not mate in the past to share DNA.
Yeah, and there's other, that was just one example, and that's what they call convergent evolution, but it's just explaining the phenomenon. It doesn't tell how it happens. And there's numbers of examples. I just happened to pull that one up, that widely related species have settled the problem that they needed to settle with the same way of settling it. And there's no relationship between the two organisms that have arrived on these solutions.
Okay, it says, next question says, you showed a diagram comparing similarities among several species for the same protein. If horizontal gene transfer does take place, it seems strange that lots more mutations or divergence takes place after the transfer. And
well, if it's an important protein, if it's an important thing, if there's some mutations that happen in any one of those in the DNA or in the RNA, it mostly be in the DNA. If there's a mutation in it, essentially, if the very important proteins have to be exactly alike, because if there's a mutation, the organism dies. And so they're maintained as exact replicas because they're so important. Some of the RNA, the things that make up the ribosome, those proteins in the ribosome are almost identical because they're so important that if they're not exactly the same, the organism will die. With just one mutation, it'll die.
So the ribosome is where proteins are manufactured, correct?
Yes. Yes, it's a ribosome is consisted of ribosomal proteins and our RNAs that are ribosomal RNAs, and it's so important that that the ribosome is an incredibly complex organelle that reads the DNA, brings in the proper amino acid, hooks it together properly in the right order. And it's a really, really important organelle. There's numbers of them. And if something's wrong there, essentially the organism dies. Or at least that cell will die if it's a multicellular organism.
Right. So they're just, yeah, some functions are common and it's like they have to be common.
Yeah, that gene I showed you that had the amino acids. Let me see if I can get back to that. Where was it? Yeah, this is a gene that is connected to, if there's a mutation in there, it's connected to cancer causing. This came from a carcinogenic sequencing of that particular protein. And so if it causes cancer, because this particular protein isn't functioning properly, it will not do its job properly and you get cancer and then the organism dies.
Okay, I have a question here. Let me see who this is from. Sean, go ahead and unmute and ask your questions. The one, I have a similar difficulty with gene duplication. Once a gene is duplicated, how is one chosen to mutate a new function and one conserves its original function? I'm not quite sure what your question is. Go ahead and unmute and ask it.
Okay, so I've read some of Michael Behe's stuff and some other things from Discovery Institute, and these molecular biologists will talk about this process of gene transfer, where in a single organism, say a gene on one chromosome that catalyzes a certain particular reaction when its gene is transcribed from the DNA, is duplicated somehow. I don't know, there's some series of enzymes that will duplicate a gene and transfer it to another part of the genome on another chromosome. How is it that these two genes that have been duplicated can follow different destinies, so to speak? One conserves its original function its protein catalyzes the original function or original reaction it had in the cell, but this new gene transcript somehow undergoes some process of evolution to acquire new function. That seems very strange to me. That seems very arbitrary to me that such a process could happen. That to me, conflicts with the standard ideas of evolution, natural selection. But how does that, how would you explain something like gene duplication if it does take place from a design point of view?
There's that is the idea behind gene duplication, is that if it's an important gene and you start monkeying with it, then if you put a mutation in, it might kill the organism. So then they hypothesize, well, and sometimes when DNA is being replicated, there will be stutters in there and there'll be duplications in there. And if it's important, the organism dies. If it's not important, that duplication gets carried along. And so then they hypothesize, well, if the gene is duplicated, then the one is doing its function and the other one can mutate and maybe come up with a new or related function because it's not going to kill the organism. There's a couple problems with that. There is sometimes organisms are very sensitive to the amount of protein that is produced. And if you have two genes producing proteins that are not regulated properly, that might hurt the organism. On the other hand, on the other side of the argument, in yeast, there is, if you, let me see, where was that? I can't remember where I had it. The yeast. Here, this is the tubulin here that pulls these chromosomes apart. And these are made of two different types of tubulin genes. One is called TUB1 and the other one's called TUB2. They sit right next to each other on a genome in the gene. And the idea was that the tubulin gene was duplicated and then slightly modified. And so now the modern day yeast have this tubulin is made up of both TUB1 and TUB2, and they coordinate to make a strong tubulin to be able to pull this chromosome apart. So there may be arguments on both sides. Yes, it looked like the TUB gene got duplicated to have TUB1 and TUB2, or is it TUB alpha, TUB beta? I forget. But on the other hand, sometimes if you have two genes, if it gets duplicated, it kills the organism. So I could see that there might be some support for gene duplication. There probably is not a lot of support for making a new unrelated function to that, because most of the time organisms are highly developed and they don't like new functions going on. It usually interferes with the biochemistry of the cells. Does that make sense?
Yeah, I think that makes sense. If I can make a comment here, in the research that I was doing basically in farming and plants and things like that, you see this phenomena going back to the horizontal gene transfer and how this information gets done. It's one thing to look at the fact that you have something like this gene transfer going on. That seems to be apparent, that's been observed. The interesting question to me is what's managing that, because you see this phenomena where a pesticide or a herbicide, herbicides in particular with plants, almost regardless of what kind they are, have a life of around 30 or 35 years, because the plants, like antibiotic resistant bacteria, learn how to manage. So you get this, Roundup right now, and you have the problem in farming of Roundup resistant weeds. And the Roundup resistance genetically is accomplished in the same way on entirely different continents. So the really interesting question is, what is managing that process? And then we had this question about here, could a duplicate gene be considered an engineering redundancy by design? I think that's an interesting question, Betsy, because if you have these things like this propagation across continents of resistance to herbicide in in crop plants, it seems that there is a mechanism there that's working the same way that can't be accommodated for, again, by horizontal gene transfer because of the distances involved.
There's plants are a lot more lenient with the ploidy of how many duplicate chromosomes. Plants can be n, if n is the number of chromosomes there are, we're 2n, most eukaryotes are 2n, and yeast can be 1n, like one set of chromosomes instead of pairs of chromosomes. But plants can have 4 pairs of chromosomes, they can have 16 pairs of chromosomes, so they're much more flexible to having multiple genes producing the same protein. They're much more resistant to, in higher organisms, in certain parts of the genome, if they're duplicated, it'll kill the organism. But plants are a lot more resistant, so there might be a lot more flexibility in duplicating the genome and then playing with the ones that aren't important, that aren't necessary, that they can come up with that because it won't kill the organism, the plants in the meantime.
Okay, so we have this additional question. So when gene duplication takes place, does the new gene not carry with it regulatory components?
If it's duplicated, it might just read right through. I'm not quite sure. There is, because there are regulatory upstream of the gene, there is transcription factor binding sites that tell it. To get a gene transcribed, it's a whole set of, I think it's like 8-10 proteins that have to land upstream of the gene at something called a transcription factor binding site. And this set of proteins put them together is called the transcription factor. And then it walks along the gene and then starts to transcribe the gene until it gets to the back end of it. And then there's a stop site, and it does its thing and puts on a poly tail on the end of the transcribed RNA, mRNA then. And so it's very complicated. And I think that when you duplicate it, it might just read through that second gene. I'm not sure. Because if it depends on if it's duplicated with the stop site or without the stop site. The stop site might be downstream of that second duplicate gene, but I'm just guessing at that.
Okay, couple more questions here.
Here, I'll give somebody else just something to think about too. I had something at the very end here. There. Those are just pictures of, it's hard to tell what's been designed and what has evolved. So I will tell you what those pictures are at the very end.
Okay, here we go. We had this question of when gene duplication takes place, does the new gene not carry with its regulatory components? I think you answered that. What do you think drives horizontal gene transfer? Are there mechanisms to facilitate it? Is there, if this is an engineered creature, is someone looking for the mechanism? What manages this? Is it? I think maybe a good way to ask the question when you're talking about horizontal gene transfer, particularly maybe if it's under stress, a situation where it's happening as a stress. And you get things like antibiotic resistant bacteria. Are people looking into what manages that process, or are they assuming that it's random, or that the antibiotic resistant bacteria in this particular case just get lucky, or are they looking for a mechanism that actually manages that process?
I think in bacteria it happens a lot, not intentionally, oh, let's share genes so it can be antibiotic, but it just happens regularly. They send out what they call pili, and they just change their genomes or pieces of their genomes with each other. And they're not as sensitive to having the proper number of chromosomes, because in bacteria, I believe it's just circular. They don't really have, they don't have a nucleus, And it's just a circular piece of DNA. So it's a lot more flexible in being able to share its genetic material.
Right.
Because bacteria
But-
aren't as sensitive to how much genetic material is in there. Whereas,
Right. I get that, but I think the question is that a directed process or is it just random? Because it seems to be a directed process, but I don't know if that's the case or not. I think that's the point the gentleman is trying
yeah,
to get to.
I have the sense that it just happens a lot in general, not necessarily, oh, I need antibiotic resistance, I better get together with this other one. I think they just generally share their DNA with each other because it's easy, and they just kind of put the pili together and run the little circular piece of DNA through the little tubes to each other. I'm not sure, I'm guessing at that, but I haven't worked with bacteria in that way. My thing was the yeast.
Yeah, so there's a question, is MPS1 a form of cancer or I guess that's, or a symptom cause? Ted, I'm not sure what you were asking there. You can unmute and ask that.
Yeah, you alluded to the MPS1 and cancer. Is it being researched as a cause of cancer and it floating around in different cells?
Well, the MPS1 gene is a controlling gene. It's monitoring, at least in yeast, it's monitoring if all the chromosomes have duplicated properly and everything's lined up properly. And so then if it's not properly lined up or hasn't duplicated, doesn't have the right number, it's got too many, it's got a 3N someplace, or if there's something wrong with one of the chromosomes, the MPS1 is a signaling, it produces a signaling protein. And if things aren't properly correct before it could divide, MPS1 shuts the whole system down in that cell. Now,
so in other words,
if it's not working properly, the cells will divide without all the chromosomes being there or extra chromosomal material there. And that's when you start having problems with cancer, if there's something missing or something broken, or it has too many. There's two genes, P51 and P21 in humans, that if either one of those is mutated, then those are such high-level controlling genes to shut down the cells. And if they are mutated, those cells will just keep dividing without any control, which is what cancer is. In fact, there was a young friend of my kids whose sister had a mutation in P21, and she died in her 20s. She had, I think, 5, 6 cancers before she eventually died, but it was because she had a mutation in one of those controlling genes. MPS1 might be something similar to that, but those are the real high level. The P53 and P21 are very high level controlling genes to kill a cell, to have it commit suicide, essentially, if there's something wrong with the duplication process. And if it's broken, the cells just duplicate uncontrollably, which is cancer. Yeah, so here's a picture. This on the left is a picture of the York Minster window, and the one on the right here is a view of DNA coming down from the top side down. So both of them, from my point of view, look designed. Are there any other questions?
If not, I have one administrative announcement. We have sort of a pent-up demand for doing more. You know, we have this mailing list. We want to give you on the mailing list the option to hear more of these. So we are in the process of planning more of these talks, perhaps one in April, perhaps one in May. So stay tuned and be watching for another email when we're ready to do the next one. Thanks for attending.